Welcome back. What I want to do in this video is go over the allosteric and a little bit of other types of regulation of the TCA cycle. And I apologize if you hear something in the background. That's my dryer going right now. But anyways, so at the end of glycolysis, recall that we had produced pyruvate. Let me get my brush. We had produced pyruvate, right? And if you need to see structures, you can certainly go back and watch the other videos. I'm just going to abbreviate things right now with their names. And the pyruvate that was produced by glycolysis goes into the mitochondria, right? And it gets consumed by something called the Pyruvate Dehydrogenase complex. And if you recall, we looked at the mechanism of it earlier in another video, and we saw the entire mechanism, how it's divided into three different enzymatic activities. There was Pyruvate Dehydrogenase, dihydrolipolysine transacetylase, and dihydrolipolysine dehydrogenase. And in the result, we produced an acetyl-CoA. So one of our products was we produced acetyl-CoA. And we also produced an NADH, although we're not going to put that here. And again, it was catalyzed by the Pyruvate Dehydrogenase complex, right? So then the acetyl-CoA went actually into the TCA cycle, and it got consumed by citrate synthase. So this enzyme right here is citrate synthase, and that was, at least in the TCA cycle, that was the first allosteric enzyme, right? And of course that formed citrate, right? Okay. The next enzyme was aconitase, right, or aconitate hydratase. So aconitate hydratase, although this particular enzyme was not allosteric, right? This was not an allosteric enzyme, so let me go ahead and X that out. This was not an allosteric enzyme. But the next enzyme that we saw was, and actually this forms something called isocitrate, right? And then that gets consumed by isocitrate dehydrogenase. So isocitrate dehydrogenase, right? And that formed what? It formed alpha-ketoglutarate, right? Alpha-ketoglutarate. And then the alpha-ketoglutarate that was produced gets consumed by the alpha-ketoglutarate dehydrogenase complex. So here's alpha-ketoglutarate dehydrogenase. And of course that forms succinyl-CoA. So here's succinyl S-CoA. And then of course we have the rest of the cycle that goes back and reforms citrate, right, by citrate synthase. So if you recall, all these enzymes that I have here, so Pyruvate dehydrogenase, citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. These are all allosteric enzymes of the TCA cycle. Now, we usually don't consider pyruvate dehydrogenase part of the TCA cycle, but it is associated with it, obviously. And these four enzymes right here, of course, three of them are in the cycle, are all allosteric enzymes, the exception being aconitate hydratase, right? So now what I want to do is I want to look at the actual allosteric effectors and make some sense of it. Okay, so let's go back and we're gonna certainly go back and watch the video on energy charge, right? But let's say I have a lot of I just had a big meal and I initially produce a lot of pyruvate, right? And the pyruvate goes through this cycle, and you fuel the electron transport chain, and after the meal, right? After the meal, you initially build up a lot of ATP, right? And in the process, while that ATP is high, you also have a lot of NADH, right? And that's produced by PDH, produced by isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, certainly. Glyceraldehyde 3-phosphate dehydrogenase produces it. There are plenty of other enzymes that produce it. Got lots of NADH. We certainly have a lot of acetyl-CoA, right? So we got a lot of high energy charge type of molecules around. And assuming that we have a lot of those around, maybe we don't need to be producing any more of them, right? So it would make sense, for the sake of energy saving, to ultimately shut down the enzymes that produce them, or at least shut down the enzymes that contribute to the production of them. Certainly some of these enzymes don't. Like citrate synthase does not produce NADH, although it contributes to the production of it by fueling the TCA cycle. Okay, so let's talk about the allosteric effectors that would be associated with high energy charge. And certainly some of them are listed right here, okay? And so it turns out that in all of these enzymes that are affected allosterically by high energy charge molecules, those molecules tend to turn off these enzymes, okay? So pyruvate dehydrogenase, let's talk about and I'm going to do the allosteric inhibitors in red. So let me write that over here. The inhibitors will be in red, and I'm going to do the activators in green. The activators will be in green. Okay. So the inhibitors of pyruvate dehydrogenase are going to be, the high energy charge molecules. So the inhibitors will be things like ATP, NADH. So NADH is an inhibitor. Also, we have acetyl-CoA, right? And there's also one other allosteric inhibitor that we didn't mention earlier, and that is free fatty acids. So let's make some sense of this, right? The fatty acids, the fatty acids will ultimately go into beta oxidation, and assuming that and actually none of the enzymes in beta oxidation are allosteric. So assuming that we load the body up with fatty acids, it would make sense that we're going to have a lot of beta oxidation, and the ultimate end result of beta oxidation is the production of acetyl-CoA. So you could almost view the accumulation of fatty acids as an indication that we're going to produce acetyl-CoA. And certainly there is an NADH also that's produced in beta oxidation. It's done by beta-hydroxyacetyl-CoA dehydrogenase. And of course then, when we have a lot of acetyl-CoA, that forms ATP. So fatty acids, they're sort of a signal that we're about to produce more ATP. So certainly, if we have a lot of them present, we can shut down pyruvate dehydrogenase, ultimately because beta oxidation will end up producing acetyl-CoA. Okay. And it turns out then that the low energy charge molecules, which are essentially the opposite molecules as these, right? The low energy charge ones tend to tend to activate pyruvate dehydrogenase. So the allosteric activators would be adenylate, also NAD+, right? NAD+. Then we also have coenzyme A, so CoA. And then it turns out that also calcium ions can do it as well. And to make sense of that, let's come over here. So let's say I'm actively contracting muscle, right? So let's say, let me do it in red. Typically they draw it in red. So here's my muscle, right? There's my muscle, right? Got a muscle, right? And if we zoom in, right? If we take this little section and we zoom in, right? We zoom in. We have this inside of the cell, right? There's the inside of the cell, right? And inside the cell, there's sort of an offshoot of the rough ER. And it's this specific type of rough ER called the Sarcoplasmic Reticulum. It's usually abbreviated SR. And inside the sarcoplasm, and actually let me designate this. This in here is the cytoplasm. This is the cytoplasm in here. And so the Sarcoplasmic Reticulum is sort of an offshoot of the rough ER. It's a type of rough ER. And inside the Sarcoplasmic Reticulum, there are calcium ions. And so during muscle contraction, calcium ions exit the Sarcoplasmic Reticulum and end up in the cytoplasm, right? They end up in the cytoplasm, right? And so it would make sense that if we have a lot of calcium ions, we're undergoing a lot of muscle contraction, right? Oops, there's a lot of muscle contraction, right? And so if there's a lot of muscle contraction, then there's a lot of ATP hydrolysis because the myosin heads, the myosin proteins that are in the muscles that contribute to the muscle contraction, ultimately with each power stroke, they burn an ATP. So high calcium tends to signal that we are in an active metabolic state. So whether we're burning, ATP in muscle contraction or whatnot, when calcium is released, it tends to signal high metabolism. And if we're in high metabolism, it would make sense that we need a lot of acetyl-CoA, right? Because the acetyl-CoA is going to make more NADH, and of course that's going to fuel the respiratory chain, right? So if there's high calcium, it probably means we're in an active metabolic state. Okay, so that of course means we need more ATP, and therefore we need more acetyl-CoA. So that will allosterically turn this enzyme on. Okay. Let's move on to citrate synthase. Citrate synthase is allosterically inhibited, again by ATP and NADH. So these two are the same, right? But it turns out that instead of being inhibited by acetyl-CoA, it's actually inhibited by succinyl-CoA, right? It wouldn't make sense if it was inhibited by acetyl-CoA, right? Because acetyl-CoA is the substrate, right? if we have a lot of acetyl-CoA, that should potentially, force the enzyme to, if there's simple enzyme kinetics, if there's more substrate, then there should be more turnover, right, by the enzyme. So it wouldn't make sense if acetyl-CoA was the inhibitor, but it turns out that down the chain, down the TCA cycle, that succinyl-CoA is the inhibitor. So succinyl-CoA, I'm going to abbreviate SUCC-CoA. Succinyl-CoA allosterically inhibits citrate synthase, and also the other allosteric inhibitor is citrate itself. So citrate, which is the product of citrate synthase, obviously, can come back and allosterically inhibit citrate synthase. So if we've got a lot of citrate floating around, it probably means that citrate synthase is really active, right? And quite frankly, I don't know of any other enzyme that produces citrate, so if there is a lot of citrate, citrate synthase is probably too active, right? And so if it's too active, then we can afford to shut citrate synthase down. And the same logic can be applied to succinyl-CoA, right? If there's a lot of succinyl-CoA, it probably means that there's a lot of citrate synthase activity. And if there's too much succinyl-CoA, then we can afford to shut the enzyme down, right? And then onto the activators, the allosteric activators. One of the low energy charge activators turns the enzyme on, and that's adenosine diphosphate, right? Okay. Aconitate hydratase is not allosteric. It does have some regulatory activity in iron metabolism, but it's not an allosteric enzyme as far as we know. It's Michaelis-Menten. So let me write that over here. This is a Michaelis-Menten enzyme, which means that it doesn't have allosteric sites. There's no effectors that can bind to it and activate it or deactivate in any way. Okay, but the next allosteric enzyme is Isocitrate Dehydrogenase, right? And it turns out that this enzyme is allosterically inhibited by ATP, right? If we've got a lot of ATP floating around, it probably means we have a lot of NADH, a lot of high energy charge molecules, and it probably means that isocitrate dehydrogenase is running full force, right? So we can, if there's lots of ATP, we don't need any more of it. So it would make sense to save, if we don't want to build up too much of it, right? So for that reason, we shut isocitrate dehydrogenase down. Okay. Now it turns out that the allosteric activators are ADP, so low energy charge tends to turn the enzyme on. If we have lots of ADP built up, right, we probably have low ATP, so we need to make more ATP. So this enzyme will turn on to make NADH, and as a result, that will fuel the respiratory chain to make ATP. And then much in the same way that pyruvate dehydrogenase was activated by calcium, isocitrate dehydrogenase is also activated by calcium ions. Okay. And the last allosteric enzyme in the TCA cycle is alpha-ketoglutarate dehydrogenase complex. Okay, and it's going to be allosterically deactivated by NADH, and also by the product of the reaction, which is succinyl-CoA. So succinyl-CoA turns the enzyme off, and so does NADH. If we have lots of NADH present, we can afford to turn down the enzymes because we have already enough NADH, right? If there's enough of it, we can turn the enzyme down. And actually, for a lot of the same reasons as the other two enzymes, the allosteric activator seems to be calcium ions, right? For a lot of the same reasons. Now, one thing I want to show you is that if I asked you, which one of these enzymes is the most tightly regulated? And your answer would be what? It would be pyruvate dehydrogenase. At least allosterically, it seems to be the most regulated, right? And that makes sense, right? Once we form acetyl-CoA, it's doomed to be acetyl-CoA. Of course, it can go towards other areas, but acetyl-CoA is sort of the central molecule in all of metabolism. So once we form the acetyl-CoA, it's doomed to be acetyl-CoA. We cannot reverse it to go back to pyruvate. So the pyruvate dehydrogenase levies a lot of control as to what happens in the cell. Are we going to take the pyruvate and go backwards toward, gluconeogenesis, towards glycogen? Or is pyruvate dehydrogenase going to be active and form acetyl-CoA? And like we'll see in later videos, acetyl-CoA can fuel the TCA cycle, but it also can be used in lots of biosynthesis reactions. So in a sense, pyruvate dehydrogenase ultimately controls a lot of biosynthesis, and of course that includes amino acid biosynthesis, especially fatty acid biosynthesis and other fat biosyntheses. And the other place it can go, acetyl-CoA can go, is it can actually go into the synthesis of isoprene units. And isoprene units are essentially these 5-carbon molecules that synthesize things like cholesterol and ubiquinone and things like that. So acetyl-CoA is really the central molecule, especially in biosynthesis. So if we're going to doom pyruvate to form acetyl-CoA, and obviously the acetyl-CoA goes other places, it would make sense to levy a lot of control in that area. And certainly there is a lot of control. Now actually, what I want to do over here is I want to come over here and I want to show you that there is one other control of pyruvate dehydrogenase. And it's not allosteric. So here is pyruvate dehydrogenase, and actually let me do it like this. So let me draw 2 pyruvate dehydrogenases. And there are essentially kinases that can phosphorylate pyruvate dehydrogenase reversibly at specific serine residues, right? So if I have specific hydroxyl residue right here, there can be an enzyme, a kinase, that specifically phosphorylates it right here, right? And it turns out that when pyruvate dehydrogenase is phosphorylated, it's inactive. So it gets let me do it in red. It gets inactivated when it's phosphorylated, right? But then there are phosphatases that can take off the phosphate and return it back to the non-phosphorylated version. And in that case, pyruvate dehydrogenase is active. So what does this mean? Well, when things like ATP build up, and we saw allosterically at least, that when ATP was present, it normally turned off the enzyme allosterically. But also, when ATP is present, it can also act as sort of a covalent modulation regulator, right? Because if ATP is present, there's lots of that substrate for a kinase, right? And so the kinase now has its substrate, and it can phosphorylate pyruvate dehydrogenase. And when pyruvate dehydrogenase is phosphorylated, it becomes inactive. So this with the phosphate is inactive. However, when there's not a lot of ATP around, this sort of favors the activity of the phosphatase. And the phosphatase, of course, uses water. They're hydrolytic enzymes, and they use water to cleave off the phosphate. And of course, that leaves behind just the hydroxyl version of pyruvate dehydrogenase, and that is the active version of pyruvate dehydrogenase. And I want to be perfectly specific. It's specifically phosphorylating the enzyme 1 component of pyruvate dehydrogenase, and specifically that part of the enzyme is just pyruvate dehydrogenase. So remember that enzyme 1 was specifically pyruvate dehydrogenase. The other ones were dihydrolipolysine transacetylase and dihydrolipolysine dehydrogenase. Okay, so ultimately what I wanted to do in this video was just to explore the allosteric regulation of the TCA cycle. And recall also, I want to point this out, is that per acetyl-CoA into the TCA cycle, we get an FADH2. Of course, that really doesn't act allosterically, but we get 3 NADHs in the TCA cycle. Of course, we get 1 from pyruvate dehydrogenase, but actually in the cycle, we get 3 NADHs. And so if the TCA cycle is running full force, there's a lot of NADH being produced. And so that NADH can sort of come back, feed back allosterically, and inhibit specifically what? Citrate synthase and pyruvate dehydrogenase. And of course, alpha-ketoglutarate dehydrogenase. The only one that's not allosterically inhibited by NADH is isocitrate dehydrogenase. So I hope this video helped you get a little bit of intuition on the allosteric regulation of the TCA cycle. And if you need help with energy charge, you can go back and watch the video on energy charge. I think I might add it to this playlist as well. But ultimately, the control of most allosteric corridors, specifically this is one of them in the TCA cycle in PDH, the main control is energy charge. So things associated with high energy charge, like ATP, NADH, acetyl-CoA, those things will inhibit these enzymes. But things that are associated with low energy charge, so when we need to make more of the high energy charge molecules, those low energy charge molecules will actually turn on these enzymes. And the reason they turn on these enzymes, like I mentioned, is because these enzymes produce NADH, right? And let me show it this way. The NADH that gets produced can go into the electron transport chain, and it fuels proton pumping. And the proton pumping, as we'll see, fuels ATP synthase. And then the ATP synthase ultimately makes ATP. And of course, as we'll see later, the NADH is going to get consumed specifically by an enzyme called NADH ubiquinone-oxidoreductase. But anyways, the NADH fuels in here, and it actually regenerates NAD, right? It regenerates NAD, but the point is that when NADH is high, that causes ATP to be high, right? That causes ATP to be high, and then ATP and NADH can fuel back and allosterically inhibit these enzymes because we already have plenty of it. We don't need any more. See you in the next video.